Hello, my name is Ivan Chumakov, and today I would love to tell you about my research project, which I'm currently working on in Dr. Luda Diatchenko's lab. I'm trying to understand the relationship between physical exercise and chronic pain intensity on transcriptomic level. So, let's talk a bit about the study design. We had the blood samples, which were collected from 35 participants experiencing low back pain. They underwent 14 weeks of physical exercise. The first blood draw was done after 2 weeks of low intensity exercise, and the second was after the last exercise session. Pain ratings were collected 2 weeks before the start of the program and then 2 weeks after the end of the study. Here is another schematic to show you more clearly when the blood draws were performed. They are marked by the red arrows, so in the end of the 2 weeks and in the end of the 14th week. Okay, now let's discuss some overall information about the data which was given to us. So we knew that duration of the study was 12 weeks plus 2 to 4 weeks when the survey questions were asked to know, for example, the average pain intensity, the sites of pain, and etc. We had total number of participants of 93 individuals. We received more than 500 variables from the study, and also the description of exercises which was performed by the participants. They included sit-ups, aerobic trainings, weightlifting, running, and more. So, what major variables we ended up using in our study? We used self-reported variables, which included average 7-day pain before the study and average 7-day pain after the study. Also, we used BMI, sex, and age as a covariables. To determine the success of the exercise intervention, the statuses of resolver and non-resolver were assigned to participants based on their 7-day pain intensity score. We had 2 time points, so before 14 weeks exercise and after. Statuses were based on the pain score or 4 and more. So if the pain was 4 and more, the status of non-resolver was assigned, and if it was 3 or less, the resolver was assigned. Here is a small schematic of my analysis. So I did 5 contrasts. It's all participants before an exercise and after. I compared the expressions of on the gene level and the pathway level, resolvers and non-resolvers at time point 1, same at time point 2, also resolvers and non-resolvers over time, and non-resolvers over time. Here are some steps and tools which are used for the analysis. They included data wrangling, BASH, R, DSEQ2 package for differentially expressed genes, and FGCE package for differentially expressed pathways. Here are some preliminary results from the study. From the analysis, resolvers and non-resolvers on the gene level over time, we observed that resolvers had 1,942 genes significantly differentially expressed, while non-resolvers had only 65 of them. On the pathway level, we saw same picture. We have here almost twice as much significant pathways in resolvers compared to non-resolvers. Major differences were in inflammatory pathways, which went up over time in non-resolvers and down in resolvers. Immune pathways also showed big difference in number of pathways as well as their activity. In resolvers, changes in inflammatory pathway expression was much higher. Here is just a picture of gene enrichment comparison. On the left you can see non-resolvers, where we see the bottom mark it is at around 0.35, 0.36, while in resolvers, we have enrichment score bigger, meaning that the activity there is higher. The next picture will prove my result as well. So here what I did, I plotted the t-statistics on the gene level. On x-axis we have resolvers t-statistic, on y-axis we have non-resolvers. The most important factor is the slope here. So slope was 0.23. Each dot is the gene. The circle in the middle, you can see it indicates excluded non-significant genes. So the slope means that resolvers and non-resolvers perform the similar activities through the similar pathways, but resolvers do that approximately 4 times more intensely. Overall conclusion from these preliminary results is that active biological processes underline pain resolution in chronic low back pain patients, and active downregulation of inflammatory pathways is one of the major contributors to resolving pain. Thank you for your attention.